it's a real treat to get to share this research that Riju mentioned, and it's a privilege to be able to share it because, as you'll see, it's been a really collaborative effort between lots of involved participants. So when the pandemic happened, we, as a society, quickly recognized that maybe there was a chance that this pandemic could be over because of a technology based on RNA. And I really like this New York Times shot because it was one of the first announcements that the mRNA based vaccines from Pfizer and Moderna were being distributed across the nation. But right next to it is this picture showing RNA's chemical Achilles heel, which is that the formulations in their initial release needed to be shipped at these ultra cold shipping temperatures. So as a community who thinks about RNA and RNA design, we wanted to see if there's anything that we could do on a timescale that could have some impact on the pandemic and the release of vaccines for SARS-CoV-2 to try to design a messenger RNA that would be more shelf stable than that first generation of RNA vaccines. And the aspect of RNA that we were going to try to leverage is, so if we have a RNA sequence that exists in these triplets of nucleotides, so where each of those triplets codes for a particular amino acid in a protein. So our goal is to make a messenger RNA that will be translated to create that spike protein, which will cause the immune response and generate the stimulation that we need for a vaccine. But there's this incredibly complex code of how you go from triplets of nucleotides from these codons to a protein. And so if you do a quick calculation for something the size of the SARS-CoV-2 spike protein, there's 10 to the 633 different messenger RNAs that all would code for that same protein. And so we envisioned that it might be possible to find a messenger RNA that would, upon being translated, still give back the same protein, but could be more stable. And so the main form of degradation that the research community was concerned about for these mRNA vaccines is a break that happens in the backbone of the message. So if you cut the message and then you interrupt them and it's unable to make the spike protein. And so the chemical basis for that degradation is actually due to this OH group, which is actually the one difference between RNA and its more stable cousin DNA. And in order for the RNA to break itself, the nucleotides actually have to get into this rather strange conformation in order for the chemistry to happen. But if the nucleotides are in base pairs, they're pulled into a conformation so that they're unable to undergo this cleavage. And so this conformation is this so-called inline attack. And so we came up with this model and reasoned that the overall rate for any particular messenger RNA to degrade should be related to the overall amount that any and every nucleotide in the messenger RNA is unpaired. And so for those of you who participated in the open vaccine puzzles, at one point you might remember there was this metric called AUP, or this average unpaired probability. So we had this model for AUP, but having a model itself doesn't do you too much good. There are a lot of more steps that needed to happen to then actually design these mRNAs. A number of questions that we had were, under this model, how much stabilization could we actually expect to gain from finding a different messenger RNA? I'll talk a little bit about computational experiments that the Eterna community made happen in order to answer that question. But then even if we can design a stabilized messenger RNA, can those still function as viable therapeutics? So can they still translate protein? And then also there's evidence that long double-stranded regions and certain structures of RNA might cause immune responses. And so if we can design them, can we design stabilized ones in a range of different structures or not? And then finally, can we also get a more detailed understanding of just how RNA degrades instead of just that initial view of only if the RNA is being paired or unpaired? As you might recognize, there's a lot more detail in the particular chemistry surrounding different shapes and sequences in RNA. So to start, we're just gonna, and many of you might have seen this example before, but for those who haven't, I'm still gonna proceed with this toy example. If we take a very short protein, so this HA tag is a tiny protein that's used commonly in molecular biology. It's so short, we can enumerate all of the possible messenger RNAs that exist for it. Here, I'm showing one messenger RNA that if you were to take this protein and put it in a commercial website, if you were to order a gene and you wanted to make this protein. And so here I'm showing one structure and I'm coloring it by that model of the average unpaired probability. So nucleotides that are yellow are more likely to be unpaired, and ones that are blue are more likely to be paired. But it's short enough, I can go and enumerate every single messenger RNA that codes for this protein. There's about 25,000. If I go and select the messenger RNA with the lowest AUP, there's about a twofold decrease in the AUP or a predicted twofold increase in the shelf life. So this is all very well and good when there's 9 nucleotides, but for something on the order of 1,000 nucleotides, we can't just go and enumerate them anymore. And so this is when we had to then ask Eterna for help. So with a really amazing team of developers and scientists at Eterna and Stanford, we developed a series of puzzles for a range of different protein targets. And we asked players to design messenger RNAs that would code for these different proteins that would range from very short. There's one very short puzzle for a designed vaccine that a minimalist vaccine of sorts. We also had one for the spike RBD domain. And we also included protein targets that are fluorescent proteins that our experimental collaborators would then actually be able to test the expression of. And then finally, also there was a puzzle for the entire spike protein. And we saw that for across all of these different puzzles, Eterna participants and also the algorithm ribotree, but Eterna participants got the lowest, the best results. We saw a roughly twofold reduction in this AUP. And so a predicted twofold increase in the shelf life. And a twofold increase, the number 2 might not be that big, but when we're thinking about if a batch of vaccines would be usable, there are a number of stories where pharmacies would throw out their batches of vaccines at 5 p.m. or whenever they would close, and they'd be unable to use them the next morning. And so but a 2 to 3-fold increase in shelf life could have an impact when we're thinking about distribution at that scale. And what was also very interesting and useful to see was that Eterna participants for messenger RNAs that were highly stabilized, they could find a range of different morphologies, which was important for that question of, would we be able to find ones that might, would we be able to redesign if we needed to avoid certain structures that might cause immune responses. And finally, here I'm showing a stabilized version of the spike protein messenger RNA. We made this design and a number of others publicly available for if there, because there are many emerging parties who are trying to develop more thermostable or improved RNA vaccines, and we wanted these to be publicly available for their use. So I, up till now, was talking about results only on a computer, but we were really fortunate to have experimental collaborators who could test our predictions for the shelf lives of these messenger RNAs. Catherine Lepec and Gunwoon Bian and others in Maria Barna's lab developed this method called PersistSeq, which involved pooling on the order of 200 different messenger RNAs together and degrading them all at the same time, and then using sequencing to over time select out and monitor how much of every single messenger RNA was still there. And so together with the Barna lab, we designed a number of different RNA variants, which all coded for a fluorescent protein, for nanoluciferase. We included messenger RNAs that were varied in their 5' UTR and their 3' UTR, and then also designs from Eterna that varied the coding sequence. And what we saw pretty much straight away was that the RNAs that had their coding sequence varied showed the biggest difference in the in-solution half-life or the shelf life. This is showing what data looks like for two different designs. Yellowstone was a construct designed by Eli Fisker, and it emerged as an example of a unstructured messenger RNA that we did a lot of comparisons to. This linear design construct, I believe, was also designed by an Eterna player. And so this is looking over time. And so you can just look at how big this band here is, which corresponds to the full length messenger RNA. And you can see over time in this readout that it's degrading. But this linear design one, it remains for much longer. And so then we would quantify these, whether it's from this 1x1 or from this pooled PersistSeq experiment, and then get these measurements for the degradation rate. And so then we could use this data to actually look at how that AUP metric did. Here I'm just showing the AUP metric in comparison to another metric which had been suggested in other papers. We saw that this AUP was more correlated with our experimental data than others. But there was a question, can we do better with more data? And I'll get to that at a later point. But an exciting result was we did an experiment where we selected 24 different messenger RNAs. We selected some that came from these commercial gene vendors, which represent sort of the best of what anyone who is in the business of making protein would select from. The Eterna community voted and selected 8 different designs that represented a range of different shapes and design principles. And then we also included 8 from the algorithm RiboTree, which Stanford PhD student Christian Cho developed. So we saw in this experiment also that there was about a 2- to 3-fold increase in the shelf life of the most stable ones. And side by side, we also measured the protein expression of these messenger RNAs at 6 hours after cells had been transfected with the messenger RNA, and then also 24 hours. And it was it was an unknown, and it was a good thing to see that these stabilized messenger RNAs were also producing protein at equivalent amounts to the messenger RNAs which had come from these gene vendors, which had been optimized, one might say, to try to produce a lot of protein. So this confirmed to us that these could function as viable therapeutics in that they were still producing an equivalent amount of protein. And I also just wanted to include this data that Riju referred to. We got to test some of these messenger RNAs in collaboration with the Pfizer vaccine research development team. So this was showing 5 different mRNAs that were characterized in one of their model formulations. It wasn't the lipid nanoparticle one that the Comirnaty vaccine was in, but it is another one in common use for them. So for instance, this GeneWiz construct saw a decrease in the amount of protein activity from 0 hours to after 2 weeks of degradation. But Bugak-Mann's lost LD plus fine-tuning mod deg2 ed showed equivalent amounts of protein production after 2 weeks. So this was really a really exciting result to see. So if we go back to that initial model that we were working with of the average unpaired probability, that's what's being shown in the colors here on this structure of a big chunk of a messenger RNA. But it seems pretty intuitive that we would not expect the degradation rate at the end of this hairpin to be the same as a nucleotide that's in a multi-loop. You can think of various reasons or hypotheses. And so we also wanted to have a challenge on Eterna where we could get a more detailed description of RNA degradation rates in different structures and with different sequences. So in June of 2020, we collected about 3,000 different RNA designs. This is just a visualization kind of trying to show the distribution of sequences from different players. I sort of like this because you can see that there's this distribution of dispersity, but that there's also clusters where a particular player would include a lot of designs that were very similar in sequence space. So having variations on one particular design idea. And so researchers in the DOS lab developed a method to read out the degradation in high throughput. And briefly, the way this worked was each of these RNA molecules had a barcode attached to it that had a unique molecular identifier. The entire pool of RNAs would then be subjected to degrade rapidly, and the fragments of RNA that were remaining are then grouped together by their barcode. And based on those lengths of the remaining fragments, we could then put together a nucleotide level picture of what was degraded where. And we saw some interesting result aspects of how RNA degrades that nobody really knew before. So here I'm showing a bond-centric view of RNA. So each of these lines corresponds to a linkage, and I'm coloring the linkage by taking that roll your own structure data and averaging over all the RNAs predicted to have in this case, this would be a 1/0 bulge. So if I just look at a series of bulges where I'm increasing the one side of the bulge, you see that there's an increase in degradation as you increase this side of the bulge. And if you do the reverse, if you increase the 3' length, that there's also an increase in the degradation on that side. But if you fill out the rest of this matrix of sorts, and if you look at these internal loops where you have symmetric increases on the inside of the internal loop, those appear to remain more protected. So this was not known to us before. And we realized that this data set, there's likely very sophisticated features that we thought deep learning models might be able to pick up on. So we had formulated, now we have this data set where we have these input sequences and we have these measurements that it would be really beneficial for us and for anyone designing vaccines if we could have a model that could predict this data to a very accurate degree. And so fortunately, Riju somehow found his connection to the platform called Kaggle, which hosts machine learning competitions. And so we hosted this competition bringing in another crowdsourcing platform to try to quickly solve this question. So it was a competition that ran over a very short time span of a few months. But the people involved in it really liked it because we designed it in a way so that the second round of data, the data that would be used to test the models that were submitted, that data hadn't been collected at the time that the models were submitted. So it was truly a blind challenge. This is a figure just showing an overview of this data. So this is a heat map of basically all the Eterna submitted designs, and there was some intricate filtering that went on to make sure that the test data were constructs that were not, so there were no clusters of designs in the test data, and that was to make sure that there was as much diversity of different sequences that were in the test data as possible. So this is the results of the competition. While the competition was being run, there was what was called a public leaderboard score, and then this was the final private test set score. And this may be some of the more nitty-gritty details, but I thought that this the way that the competition was run might interest Eterna players, being this another type of game in a crowdsourced setting. So some of you may be familiar with this DEG score model that was created as a simple linear model. The teams quickly surpassed the performance of DEG score and even a boosted version of DEG score. And a lot of teams worked collaboratively and quickly arrived at a shared neural net that they then, many teams continue to use that shared kernel and build on it. And it was very similar to Eterna designs, where there's a lot of sharing and attribution, that I got this design from Ome and I did this to modify it. There similarly was a lot of collaborative work and fun that happened in making these models. And as a reference point, this experimental error bar represents the boundary of how accurate any model would be able to be. So I'm going to show some examples from the best predicted construct from the winning model. It was a model named null recurrent, and then also from worst predicted constructs. So this is predicting shape data. On the left is the prediction from null recurrent, and on the right is the blind experimental data. And so you can see that these deep learning models were able to pick up on intricacies. Like if you look at this internal loop, there's this offset that the G is less reactive than the whatever this new, I think this is also a G. And the model picked up on that the experiment also provided. The worst predicted constructs was a little interesting to see. So one of them, the model has predicted that these nucleotides in these stems will have a low reactivity, but the experiment demonstrated that they have a high reactivity, which suggests that this initial structure which was the Vienna minimum free energy structure, was likely incorrect, and that this RNA was just unfolded. And if you look at this other one, the model predicted that these nucleotides in the internal loop would have a high reactivity, but if you look at the actual experimental signal, it looks like, again, the starting structure, which the null recurrent model used as an input, was incorrect, and that in nature, the RNA actually had a stem here. So as a final test, we wanted to see how these models did in predicting the degradation of these full-length messenger RNAs. To do this prediction, because it's unlike the role your own structure data, where we had one number per nucleotide. Here, we only had one number per entire messenger RNA. So this involved summing and a normalization step. But it was very promising to see that two of these winning models had higher correlations for all of these persist-seq data, much significantly better than either the AUP or the DEG score model. And so if coming back to these questions that we had about this work, the first, how much stabilization can we expect to achieve? So we saw that for any messenger RNA, we could, just through sequence design, expect to see roughly a 2 to 3-fold increase in the shelf life. And we saw, thanks to our experimental collaborators, that they can function as viable therapeutics. And then finally, we also saw that by this high throughput data collected through, thanks to the Eterna community, and then these deep learning models, which were very quickly developed thanks to the Kaggle community, that we can indeed get a more detailed understanding of just how the RNA degrades. And so with that, I'll stop and want to acknowledge the many people and parties who made this possible, and I'll gladly take questions.